going over practice genetics problems. And before we actually get into doing the genetics problems, what you're going to need to follow along with me is a pencil. You'll also need a calculator. And then you also want to make sure you printed out the practice problem set. And this is available online on D2L. So once you have all these things, then we're ready to get started. And before we actually go into solving these problems, I want to talk about why it's important to understand how to solve genetics problems. One reason this is really important is it allows us to predict the probability that our offspring will have a certain trait. So if two individuals, let's say one person has a certain disorder and they marry somebody that doesn't have that disorder, and they might want to know, well, what is the probability that if they have a child, that child will have that disorder? By doing these Punnett squares, doing these genetics problems, we can understand or figure out what the probability is that their child will actually inherit a certain disorder. This is true for any kind of trait. So this could be even, let's just say you both have freckles and you want to know, well, what's the probability our child will have freckles? Well, you can use these Punnett squares and you can actually figure that out. So we will actually work through a bunch of these different genetics problems, and you'll be able to see how we can predict the probability that the offspring will have a specific trait. Hi guys, in this video I'm going to talk about how to do simple monohybrid and dihybrid crosses. I apologize, I do have a really sore throat, but I really want to get this video done for you, so you're just going to have to bear with me here. So one thing that's really important to understand before we do these monohybrid and dihybrid crosses is this law of segregation. So we know that each individual has two copies of a gene that controls for every physical trait that we have, whether that trait is a disease or some part of our physical appearance. So we have two copies of this gene. However, whenever we create gametes, such as our sperm or our egg, only one copy of these genes will go into each gamete. So we have two copies of a gene, and only one of them will go into our gametes. Which one of those two will go into our gametes is actually just based on chance. There's a 50% chance that your gamete will get one of those copies, and there's a 50% chance that they would get the other one. So because this is just random chance, and we know that the chances are 50% that our gamete would get either one of these, we can use this probability to figure out the probability that our offspring would get one of those, which copies of those genes. So for example, if a parent was heterozygous, we know that they have a big A and a little a. Well, there's a 50% chance that their gamete would get the big A, and there's a 50% chance that their gamete would get the little a. However, there's a 0% chance that they could get both the big A and the little a simultaneously, because these separate during the formation of gametes. And it will always be this way. Your gamete can either get one copy or the other copy of that gene. And when we have two copies of a gene, we use the same letter to designate that they are both copies of the same gene. Remember that a big A means that it's a dominant variation or a dominant allele, and a little a means that it's a recessive variation or a recessive allele. And the only two possible variations of a gene that they could have is a big letter or a little letter. Okay? So let's look at an example here. So example 1 on your practice set, it says, if a parent is homozygous dominant for a trait, what is the possible gametes that parent could make? Well, if they are big A, so these are their two copies of the genes that they have for this trait. Well, the only thing that they could give their gamete would be a big A. So there's only one possible gamete that this individual could make, one possible egg or sperm. So the answer to this would just be one possible type, only having a dominant allele in it. Let's look at example 2. So what if a parent instead was
heterozygous. So they would have a big A and a little a. So these are the two copies of the gene they have for a specific trait. What are the possible gametes that this parent could make? Well, this parent could give their child or create a sperm or egg with a big A, or they could create a gamete with a little a. And there's only two possibilities here. Again, a big A or a little a. So again, remember this word, what I'm drawing here in the circles are the possible gametes. And another word for gametes are sperm or egg. Okay, so let's do one more example here. If a parent is homozygous recessive for a trait, what are the possible gametes they could make? Well, so that means if they're homozygous means the same, they have the same alleles. Since they're homozygous recessive, that means that they have the same alleles and they're both recessive, so they're both tiny. And the only thing this parent could give to their offspring, the only gamete they could make, would be one that has a little a in it. With this understanding, it's going to be super, super easy for you to do any type of monohybrid cross, because we're always just going to follow the same steps over and over again. So let's attempt our first monohybrid cross. So example 4, it says tall plants. Oops, let me go back here. It says here that tall plants are dominant to short plants. So that means, so here we're looking at the trait for plant height. And there's two variations in plant height. They could be tall or short. So these are the two different variations of a gene we could have. Okay, so we could have a gene that causes them to be tall or a gene variation that causes them to be short. So the first thing you always want to do is make a key. This is a very, very important step. It doesn't matter if it tells you it in the description. A key is vital always. So big T equals tall because it's the dominant trait. And little t equals short. What are the possible phenotypes and genotypes of the offspring? Remember, phenotype means physical appearances. Genotype means the genetic makeup. So what are their two possible copies of, what are the possible copies of the genes that they could have in the offspring? So they want to know what the possible offspring would be if there was a cross. A cross, this word just means mating, okay? That's all that means. So if there was a mating between a true breeding tall plant and a true breeding short plant. So if you remember the important genetics terminology, true breeding just means that they have two of the same type of gene and two of the same variations of that gene. So if they're true breeding for tall, well, if we look at tall, that means it's caused by big T, so they have two big T's. So one parent is big T, big T. And again, it's always important to write down the parents next. And then if that parent is being crossed with or mated with a true breeding short plant. And we know short plant is caused by little t, so if they're true breeding, they have two of the same type of genes or type of alleles. Alleles are the term meaning the variations of the genes. So both of their alleles here are same. This individual has two of the same alleles. But notice each parent has different types of alleles. This one has big T's and this one has little t's. So we want to know the possible offspring they could make. Well, this parent here could only make one type of gamete, and that is a big T. This parent here can also only make one type of gamete, and that's a little t. So these are the parents up here. These are the possible gametes. When you do a Punnett square to figure out the possible offspring, you'll just take the first parent write their possible gametes up on the top here. And then down along the left side, you want to write the possible gametes that can be produced by the other parent. And we only have one here. And that's a little t. Then we just create a box. And what this box represents is the possible offspring that could occur based on these two gametes fertilizing each other. So if these gametes fertilize each other, then the offspring would be big T, little t. And this shows you that there's only one possible type of offspring that these two parents could create. So to answer the question, all of these offspring, what would they look like? Well, we know that if they have a capital letter, then it doesn't matter what the other allele is, they'll always express the capital letter. So this could be a little t or a big T. As long as they have one capital letter, they'll be expressing the dominant trait, which is tall. So all the offspring would be tall. And if we look at the genotype, what genetic makeup they would be, well, they would all be heterozygous here. So in this case, all tall, and it's really hard to write with this pen, all heterozygous, or in other words, all big T, little t. Okay, that's your first simple monohybrid cross. Okay, let's now do example 5. So this starts off just like the other one, where big T equals tall. And because it says tall plants are dominant to short, so short would be a little t. Now it says if two heterozygous individuals for Dihybrid cross, so both of our parents are heterozygous. We know hetero means different, so they're going to have two different size alleles, a big T and a little t. And the other parent's going to have a big T and a little t. So these are our parents here. And wants to know what is the probability their offspring will be tall. So we know each individual, each parent's going to have 2 copies of a gene. And the first step we're always going to do is figure out what gametes they can make. So this individual could make a gamete with a big T or a gamete with a little t. This individual here could also make a gamete with a big T or a gamete with a little t. Now that we know what gametes they can make, we can look at what possible offspring they could make from different fertilization events. So this parent could give a big T or a little t to their offspring. So we'll write their possible gametes on the top here. This parent also could give a big T or a little t, so we're going to write their possible gametes down the left side here. So big T or a little t. And now we're going to draw a box. Each gamete gets its own row and its own column. And now what these different boxes represent are the different possible offspring. So what could go here is what's going to go here is the fertilization event between this one and this one. So this offspring would be a big T, big T. This offspring would get a big T from this parent and a little t from that parent. Whereas this offspring would get a big T from this parent and a little t from the other parent. Now, one tip that I have is to always write the capital letter first. So every time you have a big letter and a little letter, I put the capital first. It just makes it easier to see patterns. Therefore, I know this possible offspring is the same as this possible offspring here. And then this one would be a little t and a little t. right, so the question again is asking us, what is the probability their offspring will be tall? So let me change my pen color. We know that tall, if we go back to our key, is caused by the big letter. So it's caused by a dominant allele. And we know that a dominant allele will always be expressed or shown if it's present in an individual. So what we're looking for in the offspring is any offspring that has a big T. It doesn't matter what the other allele is. They just have to have one big T and they will be tall. So if we look here, would this offspring be tall? Well, definitely that's all they got is a big T, big T. What about this offspring here? Yes, they do have a dominant allele, so this offspring will also be tall. This offspring down here will also be tall because it is big T, little t, and this one will be short. So how did we figure out what the probability is? Well, there's 4 possible offspring. There's 4 possible different fertilization events. And 3 out of 4 would be tall. So our probability is 3 out of 4. If we use our calculator, we know 3 divided by 4 equals 0.75. And if you wanted, so one possible answer you could say is 3/4 of them. But you could also represent probability as a percentage. So we know 3/4 equals 0.75. To get the percent, you just do 0.75 times 100, and then that would equal 75%. So what does this tell us? That if these 2 parents mated, there's a 75% chance that their offspring would be, if they had a single offspring, would be 75% chance that that offspring would be tall, and only a 25% chance that they would be short. All right, so let's do example 6 now. In a species of plant, tall plants are dominant to short. Same thing again. So this is easy. We just keep doing our key. Big T equals tall. Little t equals short. I'm so happy. I've been trying to make this video for so long, and my computer's too slow, and it just wasn't working. So I'm using a faster computer now, and I can actually write really fast, and it's so nice. So let's see. If a heterozygous individual was crossed with a homozygous dominant individual, how many offspring out of 300 offspring would you expect to be heterozygous? Now, right away, if you read that whole paragraph or those whole 2 sentences there, you probably would get overwhelmed if you're not comfortable solving these problems. So what I recommend anytime you're doing any of these genetics problems is take it one step at a time. Start at the beginning and just work your way slowly through the problem. This is true with any type of math problem. Really, you just got to break it down and it's not going to be too bad. But if you look at it as if you just look at it all at once, you're going to get overwhelmed and you won't even know where to start. All right, so always draw your key here, and then draw what the parents are. Well, here it's telling us that it's a heterozygous individual that's being crossed with a homozygous dominant individual. Well, we know heterozygous means they have two different letters, or different types of letters. And when I say that, I mean big letter and little letter. Okay, so they're going to have a capital T and a lowercase t. And that's being crossed with somebody that's homozygous dominant. So homo means same, and both of them are dominant. Well, it's going to be the big letter, right? So big T, big T. So this individual, this parent, since they're heterozygous, big T, little t, they're tall. This parent, since they have both of the dominant alleles, they're also tall. We want to know how many offspring out of 300 would we expect to be heterozygous. So we just solve this like we did before. We just figure out the possible gametes. Well, this parent could make a big T, or they could give their child a little t. So they could make 2 possible gametes. This parent could only give their offspring a big T. So once again, remember the law of segregation. Every individual has 2 copies of a gene, as you can see in the parents, but each parent can only give 1 copy of their genes to their offspring at a time. So this parent could only give a big T or a little t. This parent could only give a big T because that's the only type of allele they have. So let's draw our Punnett square. We put the type of gametes from this parent on the top, big T or little t. The type of gametes from this parent on the side. Draw your box. Each gamete should get its own row and column. So this box just has 2 squares here. And then we just look at our possible fertilization events. If this gamete and this gamete fertilize each other, we would have a big T and big T to make our offspring. And then if this one and this one fertilize each other, we would have a big T and a little t. And those are our possible offspring inside these boxes. Well, the question is asking us how many out of 300 offspring would we expect to be heterozygous? Well, let me change my pen again here. Oh, no. Oh, it disappeared. All right, let me write it real quick. All right, so now that I got that rewritten again, we're looking for how many we would expect to be heterozygous. So how many of these offspring here? Well, is this one heterozygous? No, it has 2 big T's. We're looking for one that has 2 different types of alleles. So here, this one does, big T and little t. So we have 1 heterozygous offspring. So in order to figure out how many out of 300, it's really easy. We know that 1 out of 2, or half of them, would be heterozygous. In order to figure out how many out of 300, you just do 1/2 times 300, and that would equal 150. And what that tells you is if a heterozygous and homozygous dominant individual mated with each other, and they had 300 offspring, we would expect that 150 out of 300 of those offspring would be heterozygous. That would be their genotype. All right, let's look at example 7. Now, this time we're looking at a different type of trait because we're looking at humans. And we're actually looking at a disorder that causes individuals not to have any pigmentation in their skin. So here it says, in humans, albinism is recessive to normal pigmentation. So right away, let's start writing our key. So we know that albinism is recessive, so that's going to have a little letter, and you can use whatever letter you would like. I'm going to use N. So a recessive allele causes albinism. So if you have the dominant allele, that means you'll be normal, or you won't be an albino. So it says if a man who is a carrier, so remember a carrier is someone who carries the recessive allele. And they're called a carrier because an individual can be a carrier of a disease, but since they're a carrier and the disease is caused by recessive allele, they don't express it. They don't show that trait. However, they can still pass that on to their offspring. So that's why they're called a carrier. So a man who's a carrier for albinism, that means he's big N, little n, because he's heterozygous. So here he's carrying the recessive allele. Mates with a female that is an albino. So she is an albino. So if we look at our key, albinism is caused by recessive allele, so a little n. So she would be little n, little n. What is the probability that their child will be an albino? Well, to solve this, we just do what we did before. What are the possible gametes this individual could make? Well, they could make a big N or a gamete with a little n. What are the possible gametes that this female could make? Well, remember, she can only give 1 allele to her gametes, and she only has 1 type of allele. So the only type of gamete she could make is a little n. What is the probability that their child will be an albino? So let's do our Punnett square. Let's put the possible gametes from this parent across the top. This parent can only make one type of gamete, so we're going to put it over here on the left side. And then we're going to draw our box. And then we're going to look at our possible offspring. So if this gamete fertilized that gamete, we would get big N, little n. If this one fertilized that one, we get little n, little n. And we want to know the probability the child will be albino. Go back to our key. Albinism is caused by the little n. The only way you will show the little n, the possibility, is if you have 2 copies of that allele. So do any of the offspring have 2 copies of the recessive allele? Yes, this one does right here. So our probability is 1 out of 2, or in other words, 1/2. Or 1/2, you could write it this way, or 1/2 is also the same thing as 50%. 1 divided by 2 equals 0.5. Take 0.5 times 100, and that gives you 50%. So either of these would work for your answer for that. If we look at example 8, this one is a little bit different. This is what we call a test cross. A test cross is when we don't know what one of the parents' genotype is, and we have to instead look at what the offspring are to figure out what the parent is. So here it says if coffee plants dwarfism, let me go back here. Dwarfism is due to a recessive allele. So that means we got to do our little key. So I'm going to use the letter D. So dwarfism is caused by recessive allele, so that means a little letter. So that means if you have the opposite, a dominant allele, you would be normal, right? Or the plant would be. An accidental pollination of a dwarf plant produces a set of offspring that includes both dwarf and normal-sized coffee plants. What is the genotype of the male that provided the pollen in the cross? Well, so we don't know the genotype of the male, but we do know that he pollinated a dwarf plant. So do we know the genotype of that plant? Well, if we look down at our key, dwarfism is caused by a recessive allele. So the only way that that plant could be a dwarf is to have 2 of the recessive alleles. So we know the one parent has to be little d, little d. However, the other parent, we don't know what they are. So here we could go about and just set up our Punnett square like we did before. What type of gamete could this parent give? Well, they could only give a little d to their offspring. This parent we don't know, so they could give a question mark or a question mark. So what I'm going to do here is I'm going to just set up my Punnett square. This parent could give a d. This one could give a question mark or a question mark. I'm going to set up my box just like that. I know this parent would give a little d to both of their possible offspring here, but we don't know what this other one is. So let's look back up here at the question. It says, or the statement up here says that their offspring included both dwarf and normal-sized coffee plants. So do we know what this other allele would have had to be to make a dwarf and normal-sized plants? Well, let's think about the first one first, dwarf. If this plant suffered from dwarfism, what would the other allele had to have been that they got from this parent? Well, to be a dwarf, you'd have to have 2 of it's caused by recessive allele, so you would have to have 2 of them to actually have that trait. So let's change the pen color here to red. So this would have to be a little d. In order for it to be normal size, well, it would have to have a dominant allele. So the allele they had gotten for their father was a dominant allele here. So in order for this parent to mate it with another parent and produce offspring that were both normal size and had dwarfism, that father had to have given them a big D and a little d, so the father had to be heterozygous. So you can see from this example how you could actually work backwards from knowing what one of the parents is, what the offspring look like, and you could actually figure out what the genotype of the other parent would have to be. So that's just the power of Punnett squares. All right, so that's the end of looking at monohybrid crosses. Now we're going to start looking at how we can solve dihybrid crosses, or in other words, looking at crossings involving more than one trait, 2 traits. All right, so let's start solving some dihybrid crosses. A dihybrid cross differs from a monohybrid cross in that a dihybrid cross involves 2 traits. So instead of just looking at height or just looking at color, we're looking at how height and color are inherited together. So we're looking at both of these traits simultaneously. Now, one important thing to remember, though, is that there's a second law of genetics that's really important. If we're looking at how 2 traits are inherited, both of these traits are going to be inherited independently of each other. So what alleles they get for height, the offspring gets for height, does not affect what alleles they get for color. So if we look at this key here, for example, this shows that tall plants is due to a dominant allele, short plants is due to a recessive allele. So we're looking at a trait of height here, and we're using one type of letter to represent that trait. But we also have a second trait here, and this is pod color. So we're using a separate type of letter. We're using G's to represent the color of plants. Green is dominant to yellow. So we look like we have one parent here who is homozygous dominant for both traits. So this parent would be tall. They have 2 big G's, and big G means green. So this is a tall green plant. And it's being mated with or crossed with a plant that has 2 little t's, which means that they are short, and 2 little g's, which means that they are yellow. So let's think of what the possible offspring are that these individuals could make. So we do this exactly the same as a monohybrid cross. We look at what possible gametes they could make. So remember, just like before, notice that for each one of these traits, they have 2 copies of a gene that controls that trait. However, similar to the monohybrid crosses, for each allele pair here, for these T's, they could only give their gametes one of those copies. So this gamete here would get 1 copy of this gene and 1 copy of this gene. Well, there's only one possible, they only have 1 variation of each type of gene here, so they could give their offspring a big T along with a big G, and that's it. So notice the offspring only got 1 copy of each type of gene. In this case, same thing, except they're recessive. The offspring could get a little t along with the little g. Okay, now there's 2 letters here because we're looking at 2 different traits at the same time. But notice there's only 1 letter of each type in each gamete. If these 2 gametes fertilize each other, what type of offspring would they create? Well, they would create an offspring with a big T, big T, little t, and a big G, little g. So notice that the offspring, again, has 2 copies of a gene controlling each trait. And in this case, all the offspring would be tall and would be green, and they would be heterozygous for both traits. So that's actually just a simple dihybrid cross we just did right there. Now we're going to look at how we can figure out the gametes for other potential parents. All right, so before we actually start doing any of these mono or dihybrid crosses, let's first practice figuring out what type of gametes each parent could make. So here we have a parent that is homozygous dominant. Oops, forgot to put the pen on. A parent that's homozygous dominant for 2 traits. What are the possible gametes that parent could produce? Well, if they're homozygous dominant for both traits, since we have 2 different traits, we know we have to have 2 different kinds of letters, so A's and B's maybe, or you could use any other 2 types of letters. So they're homozygous dominant. So homo means same. So they're going to have 2 of the same dominant alleles for each trait. So let's use A's and B's. So it would be big A, that's homozygous dominant, 2 of the same dominants. And then for the other trait, we'll use B's. You could use any letters you want. So here you can see that they are homozygous dominant for both. So in order to figure out the possible gametes they can make, well, this is similar to what we looked at before. This individual, they could only give an A type for this gene. So he would have an A. And they could give a B type for that gene. And that's it, an A and a B. And there's only one type of gamete they could make there. A gamete that has both a dominant allele and for the A trait and a dominant allele for the B trait. Let's do a more complicated example next. So now we have a parent that's heterozygous for 2 traits. So heterozygous, we know that it means they have a big letter and a little letter. So for this trait, they're going to have a big A and a little a. For the other trait, they'll have a big B and a little b. Now, this is a little more complicated because we got to figure out all the possible combinations, different combinations of A's and B's that we could make. This is where we do what we call the FOIL method. I actually just like to do this by drawing arrows. So I'm always going to pair the first letter with the first letter of the second pair. And that would give me a big A with a big B. Then I'm going to pair the first letter with the second letter of the second pair. And that's going to give me a big A with the little b. The big A got paired with the little b. Then, to figure out the possible combinations with this little letter, the second letter of the first pair, I'm going to pair that with the first letter of the second pair. And that would give me little a, big B. And then the other possible gamete would be you pair that one. You pair the second letter of the first pair with the second letter of the second pair. And you're always going to have this pattern with your arrows. Boom. 1, 2, 3, 4. So this last pairing would be little a with little b. And right there, those are the 4 possible gametes that could be made from this one parent. These are all the possible different combinations of A's and B's that you could get from that parent there. And you'll notice we'll just keep doing the same thing over and over again by drawing these arrows this way. All right, let's do one more example here. So here it says that we have a parent that is heterozygous for one trait and homozygous dominant for another trait. So if you're heterozygous for one trait, we know they're going to have a big letter and a little letter. They're homozygous dominant for the other one, so they're going to have 2 big letters. What are the possible gametes, the possible different gametes this parent could make? Well, let's do this FOIL method again. Pair the first letter. Let's look at all the possible combinations of the 2 letters we can get. We could get a big A with a big B. We could get a big A with a big B again. And then you take the second letter, pair it with both of the letters in the second pair. And both of these would be a little a with a big B and a little a with a big B. So if we look here, these 2 are the same. The first 2 are the same, right? And these 2 are the same. So the only possible different gametes they could make is a big A with a big B, or a little a with a little b, a big B, sorry. And there's only two possible gametes that this parent, different types of gametes that this parent could make. A big A with a big B, or a little a with a big B. That's the only possible combinations of A's and B's. Okay, so let's do our first dihybrid cross. So a farmer crossed a cord plant with a genotype big G, big T, little t, so he's heterozygous for both two different traits, with another plant with the genotype big G, big T, little t. So remember, we got to write our key first. So it's saying green is dominant to yellow. So green is big, and that is dominant to yellow. And tall is dominant to short, so we're going to use a different type of letter. We're going to use T's for this, for plant height. T is tall, big T is tall, little t is short. Next it says, how many plants out of 80 should the farmer expect to be green and short? So that's what we're looking for here. So next, as we did before with the monohybrid cross, we're just going to write down what our parents' genotypes are. So we have a big G, little g, big T, little t. This is one parent. Remember, they have two copies for each type of gene. And then we're crossing that with the other parent that is big G, big T, little t. All right, so this parent is green and tall. This parent is also green and tall because they have one of the dominant alleles for each trait. Now we're going to do our Punnett square. Remember, for the monohybrid cross, first you figured out what gametes each parent could produce. You're going to do the same thing here, except this time you got to look at two traits simultaneously. So we're going to look, before we knew that they could get a big G or a little g, right, when we're just looking at one trait, but now we got to look at both these. So we're going to look at the possible combinations of G's and T's that the offspring could get. But we're always going to make sure that the two alleles for one trait are separate. So let me show you what I mean again. We'll just draw our arrows. I'm going to pair up the first letter of the first pair with both letters of the second pair. So I get big G, big T, and big G, little t. Then I take the second letter of that first pair and I pair it up with both letters of the second pair. Little g, big T was my first pairing there, and little g, little t. These are my possible gametes. And we can see that this parent can make 4 different possible GT combinations. Now let's look at the parent over here. This parent, we're going to pair it up the same way. Take that first letter of the first pair and pair it up with both letters of the second pair. So we got a big G with a big T, and that's a big T there, and a big G paired with the little t. Take the second letter, do the same thing, pair it with both letters of the second pair, and we would get a big G with a big T, big G with a little t. Well, do you see any repeats here? This one is repeated with this one, and this one repeats with this one. So there's only two possible two different possible gametes, really. Big G, big T, and big G, little t. So just like I did before, I'm going to write the possible gametes along the top for this parent. And then for the other parent, I'm going to write them along the left side vertically. So the first possible gamete was a big G with a big T. And then I'm going to leave a lot of space. And then I'm going to do a big G, little t. And then I'm going to leave some space, little g, big T. And then I'm going to leave some space between that and draw the last one. So what I put here were just the possible gametes from this parent. The other parent could only make two possible types of gametes, so it's big G, big T, or they could have given a big G with a little t. Just like before, I'm going to draw my Punnett square. Each possible gamete is going to get its own row and own column, or own column. And just like we did before with the monohybrid cross, these are the possible offspring. And we're just going to bring the gametes together. So if this gamete over here on the left fertilized the gamete up here, we would get a big G, big G, big T, big T. Notice that I'm keeping the G's together and the T's together because I want to keep the two copies of a gene for a trait together. Next, we could have a big G from the parent up here and a big G from the other one. This parent can give a big T, and the other parent can give a little t. So once again, I'm keeping the capital letter first and then putting the lowercase letter second. That's why I wrote it as big T, little t. And I just like to keep this pattern always. Next one, you just keep doing the same thing. We got a big G, little g, and a big T, big T. So I'm just bringing the rows and columns together. Now I have a big G, little g, big T, little t. And just go ahead and fill out the rest of it. Big T, little t. Big G, big G. Well, now I see I'm getting a little t from both parents. So it'll be a little t, little t. And I have a big G, little g, big T, little t. Big G, little g, a little t, little t. All right, so these are 8 potential offspring here. Now, what am I looking for? I'm looking for offspring that are green and short. So I got to figure out what type of genotypes would give me a green and short offspring. Let me change my pen. Go down here. Go to red. So for an offspring to be green and short, they would have to have, let's look over at our key. This is why we do the key. Well, to be green, they'd have to have at least one big G. All right, so I'm going to draw that here just so I can remember. They have to have a big G. Does it matter what the second allele is? No, it doesn't matter. So I'm just going to put a line here because this could be a big G or that could represent a little g. Either way, you would get a green offspring. So I'm looking for at least one big G being present. Well, for them to be short, go over here. Well, short's caused by recessive allele. So in order to express that or to show that trait, they would have to have 2 little t's. So there's only one possibility here. Little t, little t. All right, so this could be a little, so it could be big G, little t, or I'm looking for big G, little t, little t. So if I look around here, do you see any 2 little t's? Everything has a big G, but now I'm looking for one. Oh, there we go. That's one. Green and short. Oh, there's another one. Green and short. Well, I want to know how many out of 80. So I do the same thing as before. How many what's my probability of my offspring being green and short? Well, 2 out of 8. So I would take 2 divided by 8. And by the way, that equals 1/4. 2 eighths equals 1/4. And then all you got to do is take 1/4 times the total number of offspring, which is 80. And then that would equal 20 plants. So my answer is 20 plants. 20 out of 80 would be green and short. So notice this was just like a monohybrid cross. The only thing that's a little more challenging is to remember that you have to do this FOIL method here at the top. You have to FOIL it, which means you have to come up with all the possible combinations of G's and T's in order to get your gametes. And then you just place your gametes along the top, get your offspring in the middle, find the ones that have the trait you're interested in. Once you get the probability, you just multiply that by the total population to get, or the total number of offspring, to figure out how many offspring would have that trait. All right, let's go on to the next thing. All right, so let's do one more dihybrid cross practice. So this one is example 13. And one way to solve this is, let's read this problem. Huntington's disease, a neurological disorder, is caused by a dominant allele. So right away, I'm not even going to keep going. I'm going to start making my key. So this disease is caused by a dominant allele, so it's going to be a big letter. So that means a little letter would be, or the recessive allele will cause an individual to be normal. Osteogenesis imperfecta, which causes brittle bones, is also caused by a dominant allele. So this we're going to use the letter B. And then that means a little b is going to make the individual not have this disorder if they had that. All right, so let's keep going. If an individual is heterozygous for both of these disorders, and they mate with somebody that is unaffected by both disorders, what's the probability of having a child with both disorders? All right, so once again, take this step by step. If an individual is heterozygous for both disorders, well, we already know it means that they're going to have a big letter and a little letter for each disorder. So they're going to be big H, little h, big B, little b. Once again, they have 2 copies of each gene, and those 2 copies are different because they are heterozygous for each trait. They're being crossed with an individual that's unaffected, so they're not affected by this disorder. Well, we know that to be unaffected, they would have to have the recessive allele. And to express the recessive allele, they would need 2 of those letters. So they would have to be little h, and little b, little b. What's the probability the child will have a disorder? Well, let's set up our Punnett square. What are the possible gametes that this individual could make? Well, that's how we always got to start. We take the big H with the big B. And then you pair that first letter with the second letter of the second pair. And we get a big H with a little b. And then we take the second letter of the first pair and pair with the first letter of the second pair. And we get a little h, big B. And then draw my arrow there. And I get a little h, little b. So these are all different possible gametes here. So this parent on the left could make 4 different types of gametes. Once again, I'm trying to figure out all the different possible HBs that we could make. Big H, big B, big H, little b, little h, little b, little h, big B, little h, little b. So 4 different types. What about the parent over here? When you're just looking at this parent, you can see that there's only one possible gamete combination that they could make, and that is to give their offspring a little h and a little b. There's no other possible combinations of H's and B's. So I just set this up like I did before. Put the gametes of this parent across the top. You want to leave a little space between each one. All right, so I just wrote those across there. The other parent can only give one type of gamete, so I'm just going to write that down over here. Each gamete gets its own row or column. Let's make it my box. And then we just bring them together. So this offspring would have a big H from the one parent and a little h from the other, and a big B and a little b. Notice I always write the capital letter first for each pair. The next one would be a big H, little b, little b. And the next one would be two little h's, a big B, and a little b. And the last one would be a little h, little b, little b. We want to know the probability the child will have both disorders. So let me go back and look at my key. It looks like both disorders are caused by the dominant allele. So that means my child would have to have a big H. Doesn't matter what the second allele is, because they will always express the dominant allele if they have even just one of them. And they would have to have a big B. The second allele for the second copy of this gene doesn't matter what it is, because once again, they just need one of each. So I'm going to change my pen color, and I'm going to circle the offspring here. So this one definitely would have both disorders. This one would have Huntington's disease, but it would not have osteogenesis. The second offspring wouldn't. Next one wouldn't. Doesn't have a big H and a big B, and the last one doesn't have a big H and big B. So what is our probability? Just 1 out of 4. You could just write it like that. 1 divided by 4 would equal 0.25. Multiply that by 100, and that would give you 25%. So you could express this either way, but there would be a 25% chance that their offspring would have both disorders. And that's it. That's how you solve dihybrid crosses, and I hope that helped a lot, and good luck solving them on your own.